Hello, higher level students. Today we're going to continue talking about the chapters we have not yet covered, or the parts of chapters we have not yet covered. This is the higher level content of chapter A31, Biodiversity, and we just started a little bit on that in class on Monday. Now we're going to continue, and we left off with this point about applying the biological species concept, which states that groups of organisms or species when they can reproduce and produce fertile offspring. And we've already mentioned some of this stuff. So asexually reproducing species are then difficult to comply with this species concept. So others are more generally used as well. For example, here, bacteria that produce asexually and that also have horizontal gene transfer. Instead, they are tricky to use this with. So we can use in here maybe morphological species concepts or phylogenetic species concepts. So in phylogenetic species concepts, you instead look at evolutionary relatedness to determine if organisms are a species or not. So the amount of DNA or protein similarity is basically what you look at instead. But that's not part of the syllabus, only the morphological and biological. But in your book you have also the evolutionary and ecological, but I don't think that's going to come up on exam. One can hope. Also in A31, we've also covered lots of this in standard level, but here they hammer home the idea that the number of chromosomes that organisms have is a shared trait within species. So all the individuals of the same species have the same number of chromosomes. And remember, we looked at the diversity of chromosomes between species, but within species you generally have the same number of chromosomes, barring some non-conjugation. No, what is it called? Damn it. During meiosis is where you would drag chromosomes in the wrong way to make, for example, polyploid organisms happen, right? But remember always that humans, 46, and chimpanzees, but also many other great apes, have 48 pairs of chromosomes, for example. And because species have different numbers of chromosomes, they are unlikely to produce fertile offspring. So even if you would like to make a human-chimpanzee hybrid, it would probably not work because we have a different number of chromosomes. Even though, of course, we've heard the stories about the Soviet Union trying to do this to get super soldiers. I'm not sure how true this is, but we've all heard about it, I guess. Or maybe not, but now you have. Now, because we've increased our ability to sequence DNA, and we're accumulating more and more sequence data from different types of organisms, it is now possible to collect DNA in the wild and then sequence this DNA and try to determine which species are living there. For example, in water or in soil, organisms leave cells, waste, and so on, where we can find DNA from them. So that's a great way to estimate the biodiversity of a soil. We'll get a larger variety of eDNA, for example. But you can also, of course, then find DNA that you have no species that matches, and that could indicate that there's a higher biodiversity or yet to be undiscovered data. So this is, if I'm not mistaken, this is partly how we estimate the number of unknown species, like that the number of species in the world can be something close to 10 million, for example, or higher. And finally, in A31, we are going to learn the idea behind a skill to identify in species in the wild. So a common way to do this, or classical way to do this, is using a dichotomous key. So in a dichotomous key, you basically open this up. You have a species that you want to identify. You open the key and you find questions that always lead to two answers, basically. And depending on which answer you choose, you get different types of follow-up questions. And finally, you will end up at a species or group, species, family, or something like that, which your organism that you want to identify should belong to. So here we have an example of plants. So we have 4 main phyla of plants. The Bryophyta, the Felicinophida, Angiospermophyta, and Coniferophyta. And these have different types of traits that we use to classify them. So this basically, most keys use the hierarchical taxonomy system from Linnaeus. But for example, then the first question we can ask ourselves to derive which plant phyla our species of interest has would be looking if it has vascularization. So if it has, so you basically get the first question: does it have vascular tissue? And if you answer no to this question, you have only one phylum that you will get to. So that means that you have a species belonging to the phylum of Bryophyta. If you say yes, you are asked to go to a new question. Does it produce seeds? If it has no seeds, then it is a Sporophyta, or it belongs to has spores instead, but has vascular tissue. Then it's a Felicinophita, so ferns and such. If you answer yes, it has seeds, you go to 3. And the next question will be, does it have flowers, or does it hide its seed, or does it have it naked? Does it have flowers? And if you answer no, instead it has pine cones or cones structures, then you will get to Coniferophyta. But if you say yes, you would get to Angiosperms, or the flowering plants. And that's basically how a dichotomous key works. Now, if we're lucky, we could go out and test this method in the following weeks. I think we're a little bit pressed for time, but otherwise in the fall. All right, and those are the how you basically come. So each come to your final species. With each sequential division, more information is revealed and specific features and blah 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 blah. It's not so super interesting. Okay, so that was it for A31, and we're now going to go into A32, which is called Classification and Cladistics. So we need to be able to classify organisms because there's so many of them and they're so diverse and show different types of features. There are some that are single-celled organisms like bacteria and amoebas here, or eukaryotes, for example. There are animals and different types of animals. There are flowers or plants of different types. There are fungus and so on. So there's an immense diversity of different species. So we need to classify organisms accordingly. And why do we want to do that? Well, just one is to explain the world, and the other one is that if we classify organisms, that will help us to do further types of studies once we made a correct classification scheme. This specific slide is not syllabus, but we've had many types of systems before. Aristotle's system is one of the oldest ones that we have knowledge of. He basically said there are plants and there are animals, and plants can be further divided into things that have soft stems, woody stems, or several woody stems, and that will be your herbs, your shrubs, and your trees. Animals, either water, land, or air dwellers, and then he also had different things like red-blooded animals and not red-blooded animals, and give live birth and do not give live birth, and rational ones like humans, and all the other ones are non-rational like animals and beasts. Now, this is syllabus. So we already mentioned the hierarchy of taxa, but now you need to understand it slightly more. Remember that this was uses the binomial system is still used. That's this system developed by Linnaeus, with the genus name with uppercase capital and species name with lowercase small letter, and usually in italics. But here the main focus is trying to understand the hierarchy of taxa and how it may be difficult to use. Remember that the hierarchy of taxa includes the domain, which has basically the largest division possible made for living organisms. Remember, we have 3 domains: eukarya, archaea, and bacteria. Within a given domain, there will be several kingdoms within. And those kingdoms could be animals or plants or fungi or so on. Within kingdoms, there are several phyla. Within phyla, there are a given phylum, there are several classes. Within a class, there will be several orders. Within orders, there will be several families. Within one family, there could be many genera. And within one genus, there could be several species. So basically, this is why it's called hierarchical, because the further up you go, you include more species. And we'll continue now with the other terms here. So it's called hierarchy of taxa. A taxa is basically a unit of grouping in systematics. So a given kingdom is a taxon. A given phylum is a taxon. And it's called hierarchical. We said this already, so write this down. As the number of species per taxonomic decreases as you go down, and they fit into one another. So within kingdom, there are several phyla. Within one phylum, many classes, etc., etc. Knowing the order of these, remember we had this mnemonic that we already brought up. Katy Perry comes over for, and you said other words for GNS. Grape soda is what BioNinja suggests. Maybe that's more classroom friendly. Now the problem with this system is that it's arbitrary and does not necessarily reflect evolutionary divergence. So, for example, if you look in one phylum, the difference between families could be different compared to another one. So the evolutionary difference between one family and to another becomes difficult. And to account for the number of divisions you can find in one phylum to another, we also have to introduce in some cases many like divisions in between these. So there could be superclasses and subclasses, or superorders or suborders, or superfamilies and subfamilies in some groups, but not in others. In some phyla, but in others, because there's way more species and a longer evolutionary history, which means that there's more differences going on. So basically, it's difficult to say something about different groups based on this system. So that's a big drawback of this system. It's still used, but we're basically now moving over into another system called cladistics, which purely follows evolution. So this is a paradigm shift in taxonomy, or classification, sorry. Which we also should know how it works. But here, if you want, you can find the hierarchical taxonomy for us. We are animals belonging to the chordates, which have I wouldn't say, it's not a backbone, actually. It's a chorda, which is basically a structure that it's close to the backbone, but it's vertebrates that have a backbone, so this is false. Mammals that have fur and milk glands. We're primates with collarbones and grasping fingers and forward-facing eyes. Hominids, primates with relatively flat faces, 3-dimensional vision. Okay, so that's actually hominids that mainly have forward face, but I think all primates have more or less forward-facing. And we're hominids with upright posture and large brain. That's in the Homo genus, and Homo sapiens is our species. So that's, if you want to, you can use this to tell people who you are. In this classification system, we've had many histories, and the syllabus wants us to bring up a revolution that happened in our ways of classifying organism in the 1990s. So these are many different types of classification schemes that have changed. So Linnaeus had just 2 kingdoms, animals and plants, but then as we discovered single-celled organisms, that led us to a new kingdom, and then understanding that there are eukaryotic species and prokaryotic species, that led to 2 empires, and within those there could be kingdoms then. And then further on there would be other types of groups as well. But this important part that the syllabus wants you to look at is this distinction between the 3 domains, so introduction of 3 domains that was done in the 1990s. This could be, I'm not sure, when we started using also something called natural systematics. Now we kind of, this old morphological system that, for example, Linnaeus used has now been kind of replaced. We want to have classification that follows evolution. And basically this idea of doing classification based on evolutionary histories is called natural systematics. In the 1990s, a revolutionary reclassification was proposed, and that's the 3-domain model. And we also brought this up in a movie we watched for The Origin of Life, where this Carl Woese character was brought up, and he and his colleagues looked at the ribosomal RNA sequences of bacteria, of basically many different types of organisms, and that's when he noticed, and his colleagues noticed, that there are basically these 3 large domains. What we previously thought were the same in this monera or prokaryotic kingdom was basically very different from each other, and that kind of made it difficult to even apply 2 kingdoms to these, and then a 3rd kingdom for eukarya or something like that, or an empire of these things, so that the bacteria and archaea, their ribosomal RNA was so different from each other that it prompted us to reclassify them, or as to be as different from each other as eukarya are from bacteria or archaea. And that led to this way of looking at the differences between organisms today, and the most fundamental difference is either you belong to bacteria or archaea or eukaryota. So basically archaea and bacteria are as different from each other as eukaryotes are from bacteria. And here we can see some features of them, but what you need to or what the syllabus brings up is that you should know that this 3-domain model was introduced based on studies of the ribosomal RNA, which suggested that the evolutionary history of these ones are so distinct from each other that we should have classified them as these belonging to these 3 different domains. And later studies also show that the archaea and eukaryotes are actually more related to each other than to bacteria. So the eukaryotes and we are basically may have originated from an archaean ancestor more likely than a bacterial one. That's neat. Now we know more. So this way of doing classification using natural classification following evolutionary relationship is that we can then predict characters of new members within that group, or members that we don't know if they have that character. So for example, if you find that a given species has some kind of medicinal compound, then related species that we later find, or that we haven't looked at yet, we can also predict that they would have that compound, so we can also try to find it in those related species. So it's more likely that organisms within that share an evolutionary history will have the same traits. And that's the advantage of this. Plus, it makes more sense as well, which I'm not sure if it's an advantage, but it's basically a better idea for how to classify organisms based on this, which also has led to some new ways of classifying organisms. But that's what you should know from this, that the main advantage is that we can predict characters for other members or new members within the group. wait, here we've forgotten to remove something. There we go. Now. Okay, here's some animations aren't working, but it doesn't matter. Okay, so when you need to know then cladistics is the new way of doing finding. It's the way of classification based on evolutionary relationship. So that's what you should write down, that cladistics is the system of classification based on evolutionary relationship. Now, here, instead of putting them in different taxonomic ranks, like genera and families and so on, we instead use unranked groupings called clades. So a clade is defined as a group of organisms with a shared ancestor and shared characteristics. So that's what the syllabus uses for defining clade. And as I have learned, clade, it also includes an ancestor and all the species that have evolved from it. So it's basically a group of species, their ancestor, and all the species that have evolved from this one. So you basically have all the offspring from one ancestor and the ancestor itself. Now, how do we determine which species belong to the same clade? And that's we use objectively, the best evidence for doing this is using base sequence of genes or amino acid sequence of proteins. But you can also use morphological traits, but then we should know that you can have analogous traits, so traits that look superficially similar between organisms because they've gone through convergent evolution, and then you can get this problem of using wings or something like that, because then you group different organisms that have no shared evolutionary history. Now, in this tree that we see here on the right, so we have this is called an evolutionary tree, which shows a hypothesis of how species are related. We can find actually two clades in this group, in this tree. All three species belong to one clade, which a common ancestor down here. So all of these would be one clade. But within here we can find the second clade of species 1 and 2 and their common ancestor. So here we have one clade, but so these species belong to two clades. This specific clade and the other clade, the larger clade. Each clade can be given a name as well, and those names may or may not correspond to the hierarchical taxonomical naming system. But that's why in some groups where have more species in them, you have to make these subdivisions to kind of account for all the different types of clades you can find in their diversity. I hope that kind of makes some sense. And note when you do when you show the evolutionary history of species, it can be done in many different ways, but these all show the exact same relationship between the species A, B, C, and D, and E. So as long as they're put together in the right kind of order, that makes it the same. You can also, so you can have them lying on the side, facing upwards, or in these kind of right angle branches, or in these types of branches instead. Actually, these are also right angles, square then, and other types of lengths. Oh, they're not all 9 degrees. Never mind. But these are all exactly the same. You can also twist around the node, so you can have B and A here, and that's still exactly the same tree. So these trees, some of them are called cladogram. So a cladogram is a diagram that shows a hypothesis of relationship between organisms. So we need to know how to read a clade, and any evolutionary history is shown as these branches. And they end, so the terminal branch is basically the last branch that leads to a given species. Usually it's the current living species, but I think you can also say an evolution terminal branch to an extinct species. Here, if we take this clade, where these evolution branches where you have a branching event, this shows a hypothetical common ancestor of these two species that are on the branches from that node. So this node represents a speciation event and the common ancestor that the speciation event happened from. So original population, daughter population A, daughter population B, that eventually led to these two different species. And this shows the unique, this branch shows the unique history for A, this shows the unique history for B, and then this branch here shows the shared evolutionary history of A and B together. And the kind of first branch of in the cladogram is called the root, and represents the hypothetical ancestor of all species shown in the cladogram. And that also continues backwards because that extends to other species that are not part of this cladogram. But ideally, there would be... So, in theory, there's of course a true tree for all species that exist that starts with Luca. That's the main root of all living species today. That's how we can connect things from the syllabus. Isn't that neat? We should also know that a cladogram only shows the relative relationships. So it's impossible to estimate, for example, if A and B are how unrelated they are to C and D in absolute terms here. It just shows order of branching events. So relative relationships. So we can't estimate when A and B separated from each other and basically how different E is from C and D versus A and B and so on. That's impossible to do in a cladogram. But there are other types of evolutionary trees that we can use. But as we said, the main idea is that we use the number of base sequences or amino acid sequences, similarities or differences between species to estimate branching in a cladogram. So here we have the amino acid sequence for hemoglobin in a few different species compared to humans. So then we can understand that humans and rhesus monkeys are placed there's one branch between them, and then there would be one branch between those two dogs, and then to birds, and finally to fish. So that would make a tree. And you can look then at to determine if something is most like a given amino acid is suggested to be ancestral, if it's the same in more species than in others, and if it's completely new, or different in one species, that's a unique mutation in one group but not in others, and so on. But then you can compare them to see how many differences there are. So humans to this is actually different species, isn't it? Yeah, it is. Well, so it's impossible. Whatever. Or not impossible, but I don't want to sit and do it now. Okay. However, this is not the syllabus, but I'll anyways bring it up. So it is possible to set a scale on the tree, on any evolutionary tree, to generate what is called a phylogram. So basically, the length of branches here show the amount of character difference, and that's not the syllabus. Here we have a cladogram and phylogram of the same data. So the cladogram only, like I said, only shows the relative difference between order of branching events. But in a phylogram, the length of the branches kind of correspond to the amount of difference between species. So here we can see that 4, 7, and 6 are closer to each other than to 5, but also that the distance to 5 is shorter than it is to 3, and to 2 compared to 1. So that's kind of, but the branch here could be longer in this way, and that would be, would make a difference. So then we can make estimates of absolute relatedness, still not syllabus. Here we have a tree with humans here here you show the scale of some amount of evolutionary distance between them. So human, chimpanzee, and to the baboon, macaque, and so on and so on. So here you see that you have different lengths here and there for different species. And they won't all end up in the same place, but these are all still living organisms today. Now, from the same data, we can generate different cladograms. It depends on the type of criteria used and assumptions that we've made. So which sequence, if there's been, which sequences are ancestral and which are not, and so on. And so if you have many trees, then you have to decide which of these hypotheses of relationship is the most likely. And we use then the concept of parsimony. And parsimony means that the most simple solution is most likely. So basically very similar to Occam's razor. The tree with with the observed sequence of variation between clades is accounted for with the smallest number of changes. So this is the key here, that it's the smallest number of changes that have evolved, which would show this tree. So the tree with the smallest number of changes is the most likely. I'm going to show you an example of this to illustrate. So here we have 4 species and 2 characters, shape and color. Now, you can then make these 3 different trees that the shape circle has evolved, that square is the ancestral, and circle has evolved once here to account for these ones. That's possible. It's not the only possibility, and that that yellow was ancestral and yellow to blue happened here in this evolutionary path to C. Here it is also instead that square is still ancestral shape and yellow is still ancestral shape, but that is instead color that evolved here once for C, but it replaced C with B, and then square to circle would have happened twice. So there we have 3 evolution changes. Here we have only 2. This is another one where yellow is the ancestral trait. Yellow to blue happened for C, and that in the evolutionary history to D we went from square to circle, and here in B we went from square to circle. Again, this is also 3. So these are 3 different evolution trees. This is the most likely, but it's not for certain that this is the truth. These are also possible, but this is the one that has the most support. So we should know that any of these trees are basically hypotheses that we can then make predictions for new. If we haven't sequenced all the data, we can sequence more data, and we would then be able to test this hypothesis based on more information. And we will predict that those would also follow the same tree. That's what we predict, that they would show the same relationship if we look at other genes and compared to the ones we have. So that's how we can test these hypotheses scientifically. So they're scientific, yay! Okay, but we're also interested then in determining when a branching event happened. To do this, we've also covered this slightly before when we talked about LUCA, but that sequence differences between extinct or current species happen gradually, allows us and the rate for how that happened allows us to estimate when species diverged. So we use then something called molecular clocks, and so we can estimate the rate of mutation in species, or how fast mutations happen, and then we can then, of course, estimate based on the number of differences how the sequence difference basically how when a branching event happened. So yeah, to generate an estimated time of divergence. But note that these are estimates. So mutation rates in populations are not always the same. They will depend on population size. So larger populations will be able to have more mutations happening. Generation time, so faster generation time, you will have more mutations basically because you have more reproduction earlier. More individuals in the same or more generations happening in the same time period. Selection pressures such as would also affect this, like stabilizing selection will remove certain mutations, so they won't show up, and that will skew our mutation rates as well. So these are all, there's always a high degree of uncertainty in these ones. But when you do this and you scale an evolutionary tree to evolutionary time, you get a chronogram. And this is also not, well, this I guess would be syllabus, but not the word chronogram, but anyways, that we can estimate these. So basically, we can, based on the differences between chimpanzees and homini and us in sequence difference and molecular clocks, we can estimate that humans and chimpanzees diverge from each other somewhere between, I think what I've seen is like 4 to 12 million years ago is the range of estimates, but that they most of them converge around 7 million years ago, something like that. And then we can do the same for all other living species and see that we have some kind of ancient primate living, or ancestor of all primates living somewhere 40 million years ago. So that's how that works. And the last thing, last thing for this is to understand that when we've now started using cladistics, we've had to then revise taxonomical or hierarchical taxonomical systems and reclassify organisms based on cladistics. So I've already mentioned this a few times before, for example, with the old world vultures and new world vultures, that those are no longer classified the same. Here's an example that they ask us to bring up, and that's a group of plants called the figworts. And as you can see, it's one of the 400 families of the Angiospermophyta phylum, so the flowering plants. And it was originally made in the late 1700s, and now includes over 275 genera and 5,000 species, or it used to up until recently. Now, so when we started looking into this group with cladistics, the cladograms that were, or evolutionary trees that were produced, suggested that these are not at all within, or do not form a single clade, but many different clades. We say that a clade is basically a monophyletic group, so it has the ancestor in all its offspring, or offspring with all species that evolved from it. And that's the only thing we should then classify as a given taxon. So you can't include groups that have different ancestry, or we cannot skip off new species that evolved from a given ancestor and place those offspring in, or those new species in different groups. That is illegal. Now we have to group only monophyletic groups, so clades. Basically, this meant that we now have made lots of differences. So basically, we made from this one family, we made two different families, and species or genera were moved to other families as well. So let's see, 50 of those 275 genera were moved to another family, and 12 genera were moved to a different family once again. 13 of them were moved to a new family that did not exist, and 2 genera were moved to a newly created family. So there's basically 5 different moves, and 2 small families were merged with the figwort family. So things that didn't think were in the figworts were now in the figworts. So yeah, made a lot of changes to how we classify organisms. So this is one example that you can use. The same is also true for us ourselves. We, like I said before, we used to think that chimpanzees were more related to gorillas than they are to us, and now we've had to reclassify them and ourselves based on understanding of our evolutionary history, that we're more related to each other. So that's how cladistics can now is starting to upend how we classify organisms. We try to use both of these systems because cladistics is very, well, both systems were quite technical, but cladistics is even more so. So we try to merge so that the hierarchical taxonomic system is kind of following cladistics. And but I think in 100 years or so, maybe everybody just will look at clades, and that's the only thing we will look at. But it's difficult to kill your darlings or remove your historical baggage, isn't it? Yeah. here we see some examples of these kind of changes. Examples of the fig worth that have now been reclassified. So these all here that are shaded have been moved into different families. But that's how it works, and that's it with cladistics. And with that, we've covered everything that we need to for the test. So we've skipped now viruses. Those will be for after the summer break, I guess. But this is will be included in your test. And I wish you all a pleasant week and take care of each other. Bye.